And now we will finally get to talk about molecular biology as part of the biochemistry course. Your book goes into probably greater detail than I will be covering. And frankly, I'm only really covering DNA, RNA, and proteins, aka central dogma, because rumors are a lot of the students in the biochemistry class do not know these concepts. However, if this was largely a repeat for you guys, this series this last week, please tell that to me via email, because I'd rather than spend energy for the next class I'm teaching, teaching more novel concepts to my students. Also, I want to point out here that we're going to go by bacteria. Not like we hung out with bacteria that much during the rest of the course, but here I specifically want to focus on eukaryotic version of central dogma, because it tends to be shoved aside when one covers these concepts in basic biology classes, because bacteria do it easier and simpler. Yet as a result, there's a lot of confusion walking about regarding how eukaryotes, with students assuming that bacteria did the same way as eukaryotes. So we will try to cover specifically eukaryotic DNA, RNA, and protein processes. Some of them are not different at all from what bacteria do, but in others, because due to eukaryotic much more complicated genome structure, a lot of the, say, RNA processing processes are completely different. So without further ado, here's our DNA and RNA assets, the polynucleotide chains with the nucleotides assembled in various shapes and forms. And this is just a slide comparing the two. I'm sure this is a review. So DNA stores protein-making instructions, and RNA is largely used to make protein, is the protein-making instruction. DNA uses AGCT, RNA uses AGCU. The reason RNA uses U, but DNA is instead chooses to use T is because T is much more stable than U. U can easily be changed to look like C, and base pair like C, and that would be a very big problem in the DNA world, because its job is to store instructions faithfully and not change. DNA in our bodies is largely double-stranded, and RNA is largely single-stranded. In fact, our cells will recognize single-stranded DNA as a virus and destroy it, and it will recognize double-stranded DNA as a virus and attempt to destroy it. DNA is only produced during cell division. The only reason DNA would be replicated, a process which we'll try to talk about today, is when the cell is planning to make two of itself. RNA is made all the time because it is the instruction for protein making, and we're making all kinds of proteins all the time in our cells. And this is how largely DNA and RNA looks like. So this is how DNA looks like. It's this goopy snot that you can easily isolate from your mouth, from bananas, from strawberries, whatever you want. The instructions are readily available online. But it's negatively charged, it's water soluble, and because DNA is so long in most of our cells, it sticks easily together and spools really well onto any kind of stick. RNA does not spool so well because it's largely much shorter than the DNA molecules. And here, the link that I clicked on is what that goopy blob of DNA actually contained. That was human DNA extracted from cheek swab, like a whole lot of cheek swabbing. And you can use the University of California Santa Cruz genome browser to look up any gene, any human gene you want, and its location on our genome. I just wanted to show you that you don't need to have any sort of special access in order to look at your DNA. In fact, now you can look not just at humans, but mice, rat, fruit flies, worms, and yeast, which are the holy five of the model organisms: mice, rats to lesser extent, but mice, fruit flies, worms, and yeast. Right. If you get a chance to look through the genome browser, you will see with your own two eyes that most of the DNA listed that you see is largely non-coding. Very little of the DNA actually carries protein-making instructions. Most of our, 1.5% of our human DNA has protein-making instructions. They bump into 2% sometimes, depending if they count RNA genes, so genes that specifically code for RNA, not protein. Majority of our DNA is actually junk, quote-unquote junk, which is repetitive DNAs of dead transposable elements and related sequences that have been hopping around our genomes and the genomes of our progenitor species since time immemorial. Some of them die, some of them reactivate. So repetitive DNA, largely transposons. In fact, 10% of our DNA is one particular transposon called the ALU element. The second largest group is introns and regulatory sequences. Actually, I think I have a slide here. There we go. Introns and regulatory sequences. Introns is a eukaryotic specificity. Bacteria don't have introns. But eukaryotic genes have exons. Each gene in the eukaryotic genome consists of exons and intron DNA sequences. Exons actually become mRNA molecules once the process of RNA transcription is done, while introns are spliced out during the process of RNA transcription, something that we will learn about when we talk about RNA transcription lecture. So a whole lot of introns, because introns can be vastly longer than exons. You can have, 100 base pair long exon and then, 10,000 base pair long intron separating two exons. Regulatory sequences are also super important because in all this mass of our DNA, largely non-coding, you have to actually be able to find the protein coding instructions and find them at the right time and place. Certain cells need to make one type of protein, other cells need other types of proteins. And the process of gene expression, what that is, how do you find the right gene in the mess that is our DNA, is assisted by regulatory sequences, largely transcription factor binding sites. So transcription factors are special proteins that can recruit RNA polymerase, the enzyme that makes RNA, to a correct location so that RNA transcription can begin there. Then we have unique non-coding DNA sequences and repetitive DNA sequences that are not related to transposons. Can be, for example, large duplications of chromosomal chunks and versions and so forth. In fact, the whole field of genomics, that's what it deals with. It deals with complicated and fascinating DNA architecture of eukaryotic genomes. Now, bacterial genomes are vastly simpler than that. They really do have, protein coding instructions and transcription factor binding sites. There's not going to be, there's not going to be vast fields of introns or vast fields of ALU transposons in simple bacteria, because their advantage is they want to replicate very fast. It takes a very long time for us to replicate our DNA before division. To add to the complexity, our DNA is not just a simple double helix going on and on and on, but it's packaged on many different types of proteins. The first type of protein that is used is our nucleosomes, or histones. So histone being the actual protein, nucleosome is the assembly with the DNA included. And the reason proteins bind DNA is because DNA carries a partial negative charge, so a histone which is positively charged, which will easily bind to the DNA and allow the DNA to be spooled. Then after the DNA has been spooled on nucleus, on histones, it's packaged into this 360 angstrom fiber whose structure is still, it's hypothesized to be just a bunch of packaged histones binding each other, but it's still being elucidated. And then we have higher and higher order chromatin structure, because in the end we have to package our DNA into these little chromosomes and keep most of our DNA fairly compact in order for it to be actually able to fit into the nucleus. Because otherwise it would be, we have a lot of DNA and it has to be very tightly packaged in order to fit into the small space that our nucleus actually occupies. Here's a picture of this whole bead on a string situation where histones have formed nucleosomes and are spooled. The DNA is spooled on them. And then you can see that there's chunks of DNA that don't have any histones on them. And are visible here as much thinner thread. And what I want to point out that even though our DNA is always counted histones, coated in histones, that does not necessarily mean that that DNA is not accessible for proteins that need to interact with it, for example, RNA polymerase and transcription factors, because histones bind any DNA sequence and they can be readily shoved around along the long strand of DNA. So you can clear up some space and you can put your transcription factor down. Here is basically what the micrograph on the previous page showed, which is the blue and pink DNA wrapping themselves around twice around histones, core particles. Now, usually when we think about higher order DNA structure, we think of chromosomes, which to most students are these X things. Now, they're not really X's, largely when the cell is not planning on dividing. They only condense into these tight stick shapes with two arms right before cell division. This picture right here, this circle right here, is actually labeled chromosomes, also chromosomes contained inside an unbroken nucleus that is not planning to undergo cell division. And each of the chromosomes was labeled with a different color still, but you can see that now they're completely unfolded. They're all wrapped around each other and jumbled together, making a lovely colorful mess. And only when the cell is ready to divide will it package them into these much more recognizable chromosomal structures. And of course, the two sister chromatids, when they are lined up on the mitotic plate, are held together by the kinetochores here that will divide when the cell is ready. And that forms the actual X shape that we're so used to. And this is a female chromosomal profile, so there's no Y here.